All right. If there's no questions, we're going to begin. Can somebody confirm that you can hear me?
Yes, we can hear you.
Thank you. You should now be able to see my screen. Today is October 17th, and we're supposed to be talking about Lab 5, Streaking for Isolation and Culture Media. You should also, this week, read the Unknown Project instructions file. You can find it on Canvas, and I think it's week 5 or 6. I'm going to be discussing the Unknown Project next week. Let me show you where you can find the Unknown Project file. The week 5 lab and the Unknown report, the Unknown instructions. So read that file. I'll talk about it next time, as well as the Unknown Project. Any question about any of that? Not next time. I'll talk about it next week. All right. So let's begin Lab 5. Lab 5, the cultivation of bacteria, culture media, inoculation, and isolation. There is some reading in the textbook, but not very much. A few pages on culture media and a few pages on obtaining a pure culture. The primary learning objectives for this lab: upon completion, you guys should, 1, be able to understand the importance of proper specimen collection and transport to the laboratory. 2, be able to explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. And 5, define the following terms: selective media, differential media, reducing media. Any questions about the objectives? All right. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they can easily dry out, and some are sensitive to the exposure to oxygen. An anaerobe does not like being around oxygen, and oxygen may kill them. And all microbes require special transport medium to preserve the specimen. Once a specimen is received in the lab for culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation. 2, isolation. 3, incubation. 4, inspection. And 5, identification. In inoculation, you just place the specimen on an artificial culture medium to initiate growth. 2, you then have isolation, that's spatially separating out microbial growth into distinct colonies. And with that, you grow them up as pure cultures. And then 3, incubation, placing the culture medium in an environment with conditions to facilitate growth. 4, inspection, upon examining the culture for the presence of microbial growth. And then lastly, identification, where you perform specific tests to ID any pathogenic microbe that is isolated. I should mention that nowadays, besides running tests to identify the species, you could also sequence the DNA of the microbe to get the species. Any question about any of that? The 5 steps will be described in detail using different types of culture media. Have I mentioned that there are different types of culture media available for use in microbiology? Have I mentioned that? Anyone? I'm not hearing anything, so I'm going to assume I haven't mentioned that. You're already familiar with general purpose media, such as nutrient agar, Na, TSA, tryptic soy agar, and then Na, nutrient agar with yeast extract. These are media that are designed to grow many types of bacteria. They're not selective, but they do not grow fastidious organisms. In microbiology, if you were to come into the lab, I could show you a textbook that's actually a little bit bigger than this here, a little bit wider, maybe a little bit longer, and it's about this thick. On each page of this book, there is one media. And the page will tell you the directions, meaning the additives for that media, and then a little bit about the media. That tells you how many different possible media we have in microbiology, because this is an awfully thick book. Right? This is actually not a book, but it's trying to demonstrate the size of that book that has a bunch of media for you guys possibly to use. We're not going to talk about anywhere near any of those media, but we will talk about a few of them. To promote the growth of fastidious microbes that have very specific nutritional requirements, we use an enriched general purpose media, such as blood agar. For example, blood agar has 5% sheep's blood added to tryptic soy agar, and then the blood supplies the nutrients that the fastidious organism requires. For example, Streptococcus pyogenes cannot grow on a general purpose media. It requires additives. And one way to give the additives is to grow the Streptococcus pyogenes on blood agar, meaning the red blood cells supply whatever is required by Streptococcus pyogenes. This we call an enriched general purpose media, or just enriched for short. There is also selective media, and these media contain chemicals that inhibit the growth of some organisms, but allow other organisms to grow. You should keep in mind 2 things when using a selective media. First, a selective media isn't necessarily perfectly selective, meaning if given enough time, an organism that's selected against can still grow. And second, even the organisms that are supposed to grow on the selective media may grow slower on the selective media than they do on a general purpose media. Any questions about any of that? There's also differential media, and these media contain chemicals that produce visible differences among different groups of organisms when grown on that same media. These differences are unrelated to how well the organism grows on the medium, but it does allow differential, dissimilar bacteria to be visually distinguished from each other. There may be different colors on a differential media. Lastly, there's reducing media, and this media promotes the growth of anaerobic bacteria. Anaerobes require low or no oxygen conditions in order to grow, and a reducing media removes the oxygen from the media. Any questions about the different types of media? All right, it's important to note that a culture media can be classified into more than one category. For example, McConkey's agar is both selective and differential. If the organism grows on McConkey's, it's likely a gram-negative because McConkey's media has additives to it that prevent the growth of most gram-positives. So if it grows, it's likely to be a gram-negative. And then if it grows on McConkey's, you can differentiate between those that ferment lactose and those that do not ferment lactose. If the organism ferments lactose, that will produce an acid which will change the pH to a purple. And here you can see both the colonies or the cells are purple, as well as the media around the cells are purple. On the other hand, if the cells do not ferment the sugar lactose, then the media will not be purple and the cells will be their normal color. So this one is either white or off-white, and it's a non-lactose fermenter. So McConkey's is both selective and differential. Any questions about any of that? Blood agar is both an enriched media, enriched general purpose media, as well as a differential media. It's enriched because a fastidious organism that requires additives will grow on blood agar, assuming the blood supplies the additives needed. Like Streptococcus pyogenes can grow on blood agar. Trying to remember another organism that grows on blood agar. It won't grow on a general purpose media. And for the life of me, I'm not remembering another one, but that's certainly one. On blood agar, it's also differential because you can differentiate the cells on their hemolytic activity. You can have cells which show no hemolysis, and that would be these cells here, which the white are the cells, and you can see the red coming right up to the cells. So the blood is not hemolyzed, and this we call gamma hemolysis or no hemolysis. There are also cells shown here that exhibit alpha hemolysis. Alpha hemolysis is also called partial hemolysis. You can notice, I'm going to blow this up a little bit, that we have the red of the blood, and then right before we get to the off-white of the cells in the alpha, we have a dark ring, a darkening of the blood around the cells. That darkening sometimes can look greenish. In this case, it looks dark, and that is from partial hemolysis of the red blood cells. It's best seen inside the alpha, where it's darker than out here. But there is a dark ring surrounding the alpha. It's a thin ring, but it is partial hemolysis, and this is called alpha hemolysis. That differs from the cells, which have beta hemolysis, and here in the white, we have the cells in a beta, and then we have the clear ring or zone around the cells, and beyond that, we have the red of the blood. The reason why it's clear is that all of the red blood cells have been lysed, and that we call complete hemolysis or beta hemolysis. You can actually hold this plate up to a light and see through the plate in the clear region. Not where the cells are, not where the blood is. Any question about blood agar? You will note usually with beta hemolysis, you will have the beta hemolysis here, and then you'll have a ring of alpha hemolysis around the beta hemolysis. Any question about any of that? It looks like I moved that, so I'm going to move it back. And hopefully I won't remember to save this file, and then it'll be fine. It should probably be a little there. This table is summarizing the different types of media, the general purpose media, promoting the growth of most organisms that are not fastidious. If they are fastidious, they will not grow on a general purpose media. And there are three, tryptic soy agar or TSA, nutrient agar or NA, or nutrient agar with yeast extract, usually called NAY or abbreviated NAY. NAYE. That differs from the differential and enriched media, which is a blood agar plate, which is TSA supplemented with blood. And it's an enriched medium because it has the blood in it. It's also differential because you can differentiate the cells on their hemolytic activity. There's also chocolate agar or CAP, which is an enriched media. It is not differential. It's only enriched. The difference between blood agar or BAP and chocolate agar or CAP is when the blood is added. In blood agar, you have to add the blood after the agar cools to around 50 degrees or cooler, and then the blood is not lysed. If, on the other hand, you add the blood as soon as the agar comes out of the autoclave, and I don't know, well, the temperature of the autoclave is close to 215. Of course, boiling is what? Close to 100. So my guess is the temperature will cool close to around 100 degrees Celsius. And then you add the blood. That will cause the blood to lyse, giving it the brown appearance. And because the plate is brown, it's called chocolate agar because it looks a little bit like chocolate, but there's no chocolate in it. It's actually blood. Anyways, the chocolate agar is only an enriched general purpose media. There's also selective and differential media. We're going to talk about three in this lab. MacConkey's, abbreviated MAC, eosine methylene blue or EMB plates, and Hectone agar or HE plate. They are all selective and differential. They are all selective for growing gram-negative, mainly gram-negative bacilli. They are also all differential on the basis of their ability to ferment the sugar lactose. There will be different colors if they ferment the sugar lactose. On MacConkey's, the sugar fermenters, meaning lactose fermenters, will be pink. On eosine methylene blue, the sugar fermenters will be a dark or greenish metallic color. And on Hectone enteric agar, the sugar fermenters will be salmon colored. They are all selective for gram-negative bacteria. PEA, or phenyl ethyl alcohol, is a selective media, and it is selective for gram-positive bacteria. It selects against gram-negatives. Trying to remember. Yeah, it gets on the cell wall of gram-negatives and prevents their growth. Mannitol salt agar is also selective and differential. It is selective for those organisms that like salt. It has 7% sodium chloride, so only if the organism can grow in 7 sodium chloride will they grow. Most organisms will not grow above 2% sodium chloride. So only organisms that are salt tolerant will grow on mannitol salt agar. It also is differential on the ability to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol, it'll be yellow in color. If it cannot ferment the sugar mannitol, it will not be yellow. The bacteria will be whatever color the bacteria normally are. Any questions about that? Now it says that the mannitol salt agar is used to distinguish Staphylococcus aureus from other staphylococcal species. Do realize that other bacteria can grow on mannitol salt agar besides members of Staphylococcus, meaning the members of the Staphylococcus family. All right, so we have inoculation. That's setting up the culture. Then you have isolation. Louis Pasteur developed the concept of apericulture. He even developed a way of getting apericulture. What he did was, let me call it paint, was he had an original solution of bacteria. And then he would take a dilution of this solution, and we're going to say 1 to 10. I don't know if I can draw that. Oh, not bad. And then he would take a dilution of that, and that would be 1 to 100. Take a dilution of that, 1 to 1,000. Take a dilution of that, 1 to 10,000. You're getting the picture. It'd make many different dilutions. I'm going to stop here, but we can continue going. He would then plate each of these in 10 test tubes. Let me write that out. And let's see if I can draw that. Yep, that one's not wide enough. Oh, well. That's a nice one. That's a little too wide. We'll go ahead and do that one just because we're going to do it. And let me see if I can cut this and copy. Oop, didn't get that. Yeah. Getting the picture? So I made 10 different tubes of each of these dilution. If ever he got a dilution where only one of the 10 tubes had growth in it and 9 of the tubes had no growth in it, he assumed correctly that one or a few cells gave rise to that growth in that one tube. And then the other 9 tubes received 0 cells, and so they did not grow. And if this was the one that had it, then these would have 0 cells in all 10 of the tubes. But this one here he would then use that growth in the one tube as a pure culture. And he was correct in most cases. That was a pure culture. However, this is a very laborious way of getting a pure culture. Let's go back to our lab. It was Robert Koch who developed an easier way to get a pure culture. What he did was he invented the streak plate method, where he simply streaked for isolated colonies growing on a plate. He reasoned that one or a few cells was giving rise to a colony. And then if he took from the cells from one isolated colony and streaked it out on a second plate and got more isolated colonies, if he took cells from that second plate, those cells went through 2 colony formation events. And those cells have a greater than 95% chance that they are pure. And that's because he streaked it out 2 times on 2 different plates. In microbiology, if you streak it out 1 time on 1 plate, about 70% of the colonies will be formed from 1 cell. And so they will be about 70% pure. But that means 30% of the colonies are formed from 2 or more cells. And it could be a clump of cells, in which case they may not be pure. So about 30% of the colonies from your first streak are not pure. And that's not good enough in microbiology. So you do need to streak it out a second time before you can say with greater than 95% assurity that, or certainty that the colony will be pure. And even then, it's not 100% guarantee, but it's very high. Any question about any of that? So Robert Koch developed a method for obtaining a pure culture that was very easy, fairly quick, although you did have to incubate the plate, and you did it a second time. And this is still the method we use today to get a pure culture. Streak it out 2 different times, getting isolated colonies on 2 separate plates, one after the other. Any question about any of that? All right, when you're streaking out a plate for colony isolation, meaning to get isolated colonies, what you do is you follow the quadrant or the, what's the 3 called? A 3-sector streak. And you streak it out 1 time in the first quadrant and then sterilize your loop. That's to remove the cells from the loop. And then you start streaking out quadrant 2. And what you do is you go back into quadrant 1 to pick up the cultures. And let me go ahead and demonstrate that, because this doesn't quite show it correctly. It's actually good right here, but everywhere else it's not really very good. So let me go through and show it. What you want to do is, we have a plate and divide it into, boy, that's not very round, divide it into quadrants. And what you can do is just mark on the back of the plate with a marking pen, a felt pen. And then you label this sector 1, sector 2, sector 3. Let's see if I can do that. And sector 4. Kind of. What you do is you streak on sector 1 20 to 30 times from a stock culture, starting near quadrant 1 and moving towards quadrant 2. You may go over where you've already streaked. Like I said, you want to streak 20 to 30 times. Oh, come on. I'm not going to draw 20 to 30 times, but you're getting the impression. As you're streaking, the cells will be diluted. So this will be highly concentrated in this region here. And then this region here will be much less, well, not much less, slightly less concentrated. Okay. And then you sterilize your loop. Usually you put it in the Bacto incinerator until it's red hot. But if you're using a flame, you put it in the flame until it's red hot. And then you cool the loop. Usually what you do is you cool it on the plate, touching it 3 times, like right here. And then you start sector 2. And usually at the beginning, you have no cells because the loop's been sterilized. And what you do is you streak into quadrant 1, where the bacteria are dilute. So you don't go further than that. You do not want to come over here where the bacteria are really concentrated. You want them where they're somewhat dilute, like this region here. And then you come into that quadrant 1, maybe 3 times to pick up cells. And then once again in quadrant 2, you streak it out 20 to 30 times, which I'm not going to do that, but you're getting the impression. And like I said, you can go over where you've already streaked. And then you move towards quadrant 3. So in sector 2, this will be the highest concentration of the cells, and this will be the lower concentration of the cells. There will be dilution as you're streaking. It'll be slight, but it will be there. And then you sterilize the loop, cool it on quadrant 3, and then begin the streak of quadrant 3 going into quadrant 2 to pick up the cells where they're dilute. And you usually go in 3 times, making sure you go into part of quadrant 2 where there are actually cells. Like if I were to streak here, I wouldn't pick up any cells. So you have to make sure you pick up cells when you go back into quadrant 2. And then you streak out quadrant 3 20 to 30 times, moving towards quadrant 4. Once again in quadrant 3, this region will be heavily, or the heaviest concentration in quadrant 3, and this will be the lighter concentration in quadrant 3. You then sterilize the loop in quadrant 4, cool it in quadrant 4, usually by touching it right here about 3 or 4 times, and then start quadrant 4, coming into quadrant 3 to pick up cells. I'll go one more. Come into quadrant 3 about 3 times. And then streak in quadrant 4 20 to 30 times. When you're doing quadrant 4, you make sure that you do not go into quadrant 1. Because if you do, you will pick up very heavy concentrations over here and then bring them to quadrant 4. So you don't want to ever go outside your quadrant except at the very beginning. Like when I'm coming here, that was pretty close to quadrant 1, but it didn't pick up any cells. And this is done correctly, although right there, I'm really close to quadrant 2, but I didn't pick up any cells. Someplace along the line where you're streaking, it might be here, it's oftentimes in quadrant 4. If it's rarely dilute, it could even be in quadrant 1. You will be plating down individual cells. So I'm going to draw it here, where we plate down an individual cell right here on the streak, and that will grow into a colony given time. And so you'll see individual cells in this region of sector 3. And that's because we're plating individual cells down. And then in this region, you'll have lots of cells growing on top of each other. So the colonies are not isolated, but in this region they are. And if you're getting isolated colonies in sector 3, sector 4 won't have any. But most of the time when students do it, it's actually sector 4 where they get the isolated colonies, but it can be any place because you're diluting the cells in each sector. And then from the beginning of the sector to the end of the sector, you're also deleting the cells in your streak. Any questions about streaking for colony isolation? The purpose of streaking for colonies is to get isolated colonies. Okay. And then once you have an isolated colony, you take the cells from that colony and streak it out on another plate to get isolated colonies. Oops, that obviously went over. I'm not going to draw this, but you're getting the idea. And if you have an isolated colony here, the cells in this colony went through 2 colony formation events. Once in this plate, and then once from this plate, because I took from one colony to set up this plate. And those cells will be over 95% pure, meaning over 95% of the colonies will be pure on this plate. Any question about that? All right. Incubation is taking the culture and putting it in an environment to cultivate microbes. We usually do that in a device we call an incubator. In our lab, we have very simple incubators that only regulate the temperature and the humidity and the time. However, you can have a more complex incubator that can regulate the amount of oxygen that's inside the incubator and the amount of CO2 that is available. In a very complicated incubator, you could even have no oxygen in the environment. There's something put in this jar that removes the oxygen, and then we can grow anaerobes. You then take the plate out of the incubator and then inspect it, looking for obvious visible colony differences between different species of bacteria. You can look at different things such as the differences in the colony, such as their size, their shape, their margin, the elevation of the colony. We'll talk a little bit more about that in this lab later. And then you identify the species by running a biochemical test. All right, here's the terms. Know the terms of this lab. Don't need to know the references. In this lab, we have some videos for you to watch. Usually there's a discussion of, I'm going to tell you that in this lab, we're going to be doing everything online. So you're not actually going to be doing this lab. What you'll do is watch videos of the lab and answer questions. There is no virtual lab for this lab. Anyways, there's a video on inoculating an agar plate using streak plate culture technique. You only need to watch this video to 4 minutes and 15 seconds into the video. And you should note that this video uses 3 sectors instead of 4 for streaking for the colonies. We then have 3 different videos, one on beta hemolysis, one on alpha hemolysis, and one on gamma hemolysis of a blood agar plate. So watch these 3 videos. Then watch the video on MacConkey's agar. Once again, on this video, you only need to watch the video for 3 minutes and 50 seconds into the video. After that, you do not need to finish the MacConkey's agar video. And then we have a video on mannitol salt agar. So watch these different videos. Let me go to my worksheet. And then answer the questions in the worksheet to Lab 5. Describe a natural circumstance where staphylococcal species of bacteria might benefit from salt tolerance. You're asking, you need to discuss someplace where naturally Staphylococcus aureus can grow in a lot of salt. And don't say in like a mannitol salt agar plate, because that's not a natural circumstance where staphylococcal species can grow. So say someplace on the planet Earth, I guess. And 2, how might hemolysis benefit a hemolytic organism? So we have a bacteria that can lyse red blood cells. How would that benefit the bacteria? 3, imagine that you have a culture containing a single bacterial species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar plates, blood agar plates, and EMB plates to help identify the unknown bacteria. That is, how would you use these plates to help you identify the species? I'm going to give you a hint. You need to discuss 2 properties to each of these plates that can help you identify the species. 4, what is the main goal for streaking for isolation? 5, what happens to the growth on the plate if you forget to sterilize the loop in between sectors when you're streaking for isolation? You need to be specific and detailed in your answer, discussing each sector. If you do not discuss each sector, I really will probably not be able to tell if you know the answer or not. So please discuss each sector. 6, what happens to the growth on the plate if you transfer from stock culture to inoculate each of the sectors? So normally when you're doing a plate, only in the first sector do you get the cells from the stock culture and then streak out sector 1. However, in this case, in sector 2, you're going back to the stock culture and then setting up sector 2. And then sector 3, you're going back to the stock culture to set up sector 3, etc. So once again, be specific in detail, discussing each sector. What happens to the growth on the plate if you transfer from stock culture to inoculate each of the sectors? So tell me what the growth looks like on each sector. Any questions about that? I will tell you that 5 and 6 will look different. But beyond that, I'm not going to tell you the answer. 7, this is a thinking question that often stumps students. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. how can you use this plate? This is not the thinking question. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer. So this is not the thinking question, okay? I've actually told you the answer to number 7. 8, in the table below are a variety of agar plates. They have different media available to microbiologists to cultivate bacteria. Denote whether each medium is non-selective, it's a general growth media, or a selective media, or a differential media by placing an X in the appropriate column or columns. You may use the table of media in the lab module to answer these questions. So nutrient agar, put an X if you think it's non-selective, put an X here if you think it's selective, put an X there if you think it's differential. For some of them, you will need to put 2 X's on the type of media, okay? And you all know that we have selective and differential media, so those will be ones that are both selective and differential. Okay, so do that for nutrient agar, tryptic soy agar, blood agar, which is BAP, chocolate agar, which is CAP, McConkey's agar, or MAC, EMB, or eosine methylene blue, hectone agar, or HE, phenyl ethyl alcohol, or PEA, or mannitol salt agar, or MSA. And then 9, your last question, which agar plate was inoculated using a streak plate technique? Plate A, plate B, plate C. You can either circle the answer or circle the plate, or you can write it out B down here or up there, wherever. Any question on the lab? All right, if not, I'll be here till 7:45. But when the last student logs out, I'll log out also. But I'm here to answer any questions and help you with the lab. So use me, you guys. I just graded, I forgot to mention that at the start. I just graded one of the labs, I think it was lab 3, and it was quite obvious that some of you did not pay attention to reading the lab if you read the lab, and then you answered the questions, and so you got a lot of the questions wrong. And you do need to read the lab to answer the questions. And then, like I said, you can use me if you're confused or you need help. Any questions? All right, go ahead and get started on the lab. I'll be here until 7:45 or until the last student logs out. Oh, if you have any other questions, you can ask them in the lab. If it's a private question, we can always wait till all other students log out of the lab and then can discuss in privacy. And you'll know when somebody logs in because the system sort of rings or beeps, whatever.